Uh, Thank you. So I am Angeliki Milioti. For today's webinar, I will be the moderator. And let's see now the agenda. Our today's webinar is entitled The Way Forward for Plant Protection, Agricultural Research in Horizon Europe. It is an online event that is co-organized by 4 Horizon Europe projects: NextGenBioPest, SAGROPIA, BIOVEXO, and VIRTIGATION. So you can see already the agenda on your screens. First of all, we are going to start with some opening remarks from Ms. Gisela Quaglia. Then we will pass to the project spotlights, which means that we will see in detail what its project is about, our 4 projects for today. Then we will have a short break, and after that, Dr. Santofanti will be the moderator for an expert roundtable discussion. And after that, we will have a Q&A session. Of course, after each presentation, we will give around 5 minutes to answer possible questions. So feel free to add your questions to the chat, and our speakers will try to answer all of them. So thank you again. Now I will give the floor to Ms. Gisela Quaglia, the representative from the European Commission, to start with her talk.
Thank you. Thank you for the introduction, and good morning to everyone. I want to extend a warm welcome to everyone that is joining us today. I'm Gisela Quaglia. I work in DG Agri at the European Commission, and I'm a research program officer responsible for the portfolio of projects on plant health. It's my pleasure to deliver the opening remarks for this important webinar on the way forward for plant protection. At a time when European agriculture is facing unprecedented challenges, our commitment to innovation and science-based solutions have never been more crucial. Ensuring a sustainable, resilient, and competitive agriculture sector requires forward-thinking approaches, and crop protection is at the heart of this effort. Through our flagship research and innovation program, Horizon Europe, we are making strategic investments that are key in addressing these challenges, with around 9 billion euros allocated under Horizon Europe Cluster 6 for the period 21 to 27 for research in agriculture, food, bioeconomy, environment, and natural resources. We are investing in the future of sustainable farming. This investment not only generates new knowledge and tools, but also ensure their application in real-world settings. Bridging the gap between scientific advancements and practical solutions is key to supporting farmers and communities with cost-effective, viable innovations. Over the past 4 years, significant funding has been directed toward plant health and crop protection, supporting projects that develop cutting-edge tools for the prevention, monitoring, control, and management of plant pests and diseases. They also aim to find safer alternatives to hazardous pesticides and promote the adoption of new technologies. Today, you will hear about BIOVEXO and VIRTIGATION, which are developing solutions to major plant pests, helping to mitigate threats to key crops, and from SAGROPIA and NextGenBioPest, which are leading the development of low-risk and biological control methods to reduce the dependency on chemical pesticides, particularly the most hazardous ones, while promoting integrated pest management strategies that are both economically viable and environmentally sustainable. Beyond individual research projects, we are also supporting systemic play-based approaches through initiatives such as the Mission Soil, a soil deal for Europe, and the Agroecology Partnership. Recognizing that one-size-fits-all solutions that do not work, these initiatives emphasize holistic, context-specific agriculture practices, By leveraging living labs and lighthouses, we ensure that on-farm innovation and testing lead to practical, scalable solutions that are tailored to diverse agriculture landscape across Europe. So, dear participants, today's webinar brings together researchers who are shaping the future of plant protection. So their work is driving forward the next generation of sustainable crop protection strategies, making European agriculture more resilient, productive, and environmental responsible. So I wish you a productive and inspired session and look forward to the enriching discussion ahead. Thank you.
Thank you very much for the insights you shared with us. And now I think we can proceed with the first session, actually, the NextGenBioPest project spotlight. So I will ask Professor John Vontas to take the floor.
Thank you very much, Angeliki. I'm not sure I cannot share my screen, so either please allow me, or you upload the presentation. For some reason, I'm not allowed to. Okay, now I'm fine.
Oh, nice.
Perfect. So good morning, everybody. I'm John Vontas. I'm based in Crete, Greece, and I'm the coordinator of a RIA project entitled Next Generation Biopesticides for the control of the most difficult to manage pests and pathogens in fruits and vegetables. This project started approximately a year ago. It's kind of a continuation of a previous project which was called Super Pests that focused on insecticide resistance and against aiming to develop means to control those difficult to manage pests. This is, however, a more mature project, which includes more mature in terms of TRLs, includes more companies. It's much closer to both application of some of the findings, as well as demonstrations to the end users, as I will explain later, via integration into IPM projects, evidence-based IPM projects. It's a large consortium with several academic and institutes and companies. As you can see in these slides, I don't read all logos, but probably you recognize some major organizations, either academics or closer to the field, such as INRA or universities or big companies which are leaders in their fields in biological control or RNAi, as I will present later. So the main objective of Next Generation BioPest, which is the Next Gen BioPest, which is the acronym of the project, is to develop innovative products and methods and integrate these with existing approaches to achieve effective, environmentally friendly and sustainable protection against specifically the most difficult to manage pests and pathogens in major fruits and vegetables in Europe, and enable uptake by the end users. So among those difficult to manage pests, I show you in this photo some entomological, main entomological pests, such as the whitefly, Tuta absoluta in tomatoes, Tetranychus urticae, an extremely polyphagous pest, as well as some major pathogens, which are also among the main aims in the targets in the project, such as mildew and other diseases. Those pests are resistant to pesticides, associated with heavy use of chemicals to control them. And there are the main problems in fruit. The more specific objectives of the projects are listed in this slide briefly. Initially, we need to set up a forum to capture the needs and expectations by stakeholders by such a large consortium and European funding. Among technical objectives, to develop automated molecular platforms and tools for diagnostic purposes, not only to identify the pathogens and pests, but also characterize them and then adjust the control project towards their profile. To develop new biological control agents and strategies to enhance their efficacy in the field in close collaboration with BIOPEST, to develop, validate and bring to the market RNA-based pesticides in close collaboration with the Green Light Bioscience, to develop, validate and bring to the market plant resistance inducers, highly selective juvenoids, pesticides which are of the class of IGR, insect growth regulators, and repurposed green chemicals that have been commercialized in other places like Australia, to optimize agronomic and ecological practice balance of agricultural production against biotic stress and enhance sustainability of IPM. Of course, to integrate the outcomes of next-gen BioPest with existing methods into novel experimentally validated IPM concepts, as well as develop capacity for demonstration demonstration fields to the end users. Of course, the analysis of social, economic, and environmental aspects of those novel biopesticides and practices, agroecological practices, is among main objectives in this project. And finally, we aim to promote the integration of knowledge, dissemination, exploitation, and uptake to end users, and this webinar is part of this specific objective. This is diagrammatic illustration of the project, as I mentioned, which shows the translation pathway actually. As I mentioned, it includes biological control agents, biopesticides, practices, new automated molecular diagnostics tools that will be developed during the project. Then they will be validated in semi-field trial in the labs and semi-field trials. The most promising, the most effective tools will be integrated into IPM projects, and the ambition is that at the end of the project, which is in 3 years from now, this is a 4 years project in total, we will have novel biopesticides fully validated in the field, novel concepts and practices in IPM, advanced research capability in Europe, and the intervention concept for alternative crops, because most of the developments are aim specific pests and crops, but they are also flexible to be adapted for other crops and pests. This is the structure, the PERT graph, the structure of the specific work packages. As mentioned earlier, it includes automatic diagnostic platforms. It includes the work package 3, enhancing biological control, led by BioPest. Work package 4, RNA-based pesticides, led by the biggest company in the field, which is based in US but has headquarters in Spain, Green Light Bioscience. Work package 5, low-risk pesticides and green chemicals to test them against the most difficult to manage pests. I should mention that we have done a lot of work on that, but it's not extremely efficient, to be honest, those green chemicals against the most difficult to manage pests. I will not show you any results on that, just I mention it. Agronomic and ecological practices to enhance the efficiency and sustainability of control. Work package 7, demonstration field, of course, integration of the outcomes into advanced IPM SEMS, demonstration fields for capacity building and training. Work package 8, led by Slovakia University, Agricultural University, socioeconomic and environmental assessment, regulatory issues, and also with a contribution from Dimitra Elgo, which is a Greek organization. Work Package 9, communication, dissemination, and exploitation. And if I have a few more minutes, I would like to briefly present some initial outcomes of the projects and what more specifically we aim for. In Work Package 3, as I mentioned, we aim to improve biological control. So we try to develop mites against spider mites in tomatoes, Typhlodromus, predatory thrips, Franklinothrips that are developed by EVIA, Spain, and BIOPEST against thrips, including several major pests in Europe. TD mites to control tomato rust mite and plant pathogens, powdery mildew, simultaneously. This is quite innovative biological control agent. The parasite Anargyrus matritensis has been included in the project. It's led by UNIC Italy against the invasive to the Mediterranean mealbug Phenacoccus solenopsis. We have also some new species, and I would like to mention that our project is flexible. So, for example, if a new pest, emerging pest, becomes very interesting in Europe during the development of the project, we will aim to develop biological control agents, or if a biological control agent evolves, for example, we try several lines that come from Africa or other places and has better fitness than others used for certain control applications, they will be prioritized. We have already publications for the certain predatory thrip, which shows the efficiency, open access. And also, in addition to producing novel biological control agents, we want to support their releases in the field based on food supplements. This is a task led by EVIA, Spain, with contributions from INRAE, UNIC, Instagro, and BIOPEST. Very important to find ways, food supplements to support the populations of natural enemies that you release, which we try to have already good fitness for those enemies in terms of adaptation to different plant varieties or even to pesticides. So we try to have as good as possible fitness for those natural enemies, as well as to support them after releasing in the field by food supplements and companion plants in some of our particularly orchards that are included in the project. And the second example that I would like to present and close with that is the trials of RNA pesticides. Particularly the mildews project has undergone an extensive trial field testing with support from next generation biopest in some cases in Europe. Very efficient control has been achieved by those double-strand RNA products and formulations similar to the conventional or application of chemicals. It's very important the formulations of those double-strand RNA apparently, and the regulatory ready formulation has been extensively tested across Europe and the US, providing consistent results. So it's we are also collaborate with Resion, which is a project which deals with the development of regulatory protocols and adapting others for low-risk pesticides in Europe. It will take some time, but we are in close association with them and try in parallel to demonstrating the efficiency, also to develop the regulatory, to be in line with the regulatory aspects of for the development of those pesticides in the market very soon. I mean, really, we hope within a year or two to have products in the European market. Those substandard RNA pesticides are also efficient in preliminary experiments against difficult to control mites, the Tetranychus urticae. This is also very promising, and there are ongoing field trials for this against those pests as well in several places in Europe. Panonicus citri, also the mite of C3 orchids populations are reduced when targeted with formulated multi-species RNA. So in this formulation, double standard RNA formulations, we try to combine active double-strand RNAs against both the Tetranychus urticae and Panonicus citri to broaden the spectrum of this of the efficiency for this application, which very promising results so far equivalent to chemical control. So finally, that's the slide that I explained earlier, how important it is to manage to generate both biological control agents with increased performance and fitness, as well as introduce novel applications such as RNAi, and integrate all those into advanced integrated pest management SEM based on scientific evidence for the pathogens and pests that are present in a crop, and also by optimizing the time of applications in a sophisticated manner. So thank you very much for your attention. I'm not sure if we have questions now or later, Angeliki.
First of all, thank you very much for this detailed presentation of our project. I don't see any questions for now in the chat. However, if any questions come up until the end of the webinar, I hope you will be here to answer all of them at the end of the sessions. So now I think you can stop sharing your screen. And nice, thank you. And now I will give the floor to our second presenter for today, the representative from SAGROPIA project, Dr. Lucius Tam. So the floor is yours to talk to us about your project, SAGROPIA.
Yep. Can you hear me okay?
Yes, we can hear you, we can see you. And
Okay.
now we can see your screen also.
Okay, and I think... Do you see full screen now?
We see the both the notes section of the presentation. Now we see full screen, yes.
Okay, very good.
Can you change the slide just to make sure that we can see it moving?
Yep. Okay.
Okay, nice.
Okay. Good morning, everyone. I'm going to tell you a little bit about what we are doing in SAGROPIA, of which the full title is Sustainable Agriculture through Novel Pesticides Using an Integrated Approach. It answers to the call listed here, which was really clearly focused on the reduction of the use of candidates for substitutions. So the call was quite straightforward. It's innovation action, and you can see here we started off last year in January, and it will end up in 28. So we are now just starting the second year. It includes 10 partners, including industry and academic partners, as you can see on the right-hand side. SAGROPIA addresses some challenges which are listed here. So it's clearly that the high yields, they so far have depended on chemical pesticides, especially also in arable crops. We see that in many cases, microbial and plant extract-based solutions cannot fully replace chemical pesticides. And there is quite some urgency to substitute or reduce the use of candidates for substitutions, which are highlighted because they are problematic. What we also started off, and this was driven by our coordinator, Günther Brader, who is in the audience, we focused on the really tricky crops, which are row, so arable crops, so large-scale crops, and in this particular context, potatoes and sugar beets, which face really high pests and pathogen pressure, which I will show in a minute. We see that, as my speaker just before has showed, in greenhouse crops, there are quite a few solutions. Scaling is really quite a challenge, and to introduce innovative pest IPM strategies is key to overcome the bottlenecks. So we need to scale up available candidates, substances. We need to optimize formulations. There is a need to understand the mode of action in some cases. And of course, there is also a need to ensure the safety and the stability of the alternatives. So this translates into SAGROPIA's objectives. We aim to integrate new if possible, low-risk solutions, biocontrol solutions in current potato and sugar beet IPM strategies. It's the aim to provide affordable replacements and hopefully to reduce the use of the chemical pesticides by 50%. And of course, we want to reduce the adverse effects on the natural resources and so forth. Initially, it was quite clear that we are aiming at oxamyl, difenoconazole, fluapicolide, metalaxyl, and others, which are highlighted as candidates for substitution. But there is, of course, behind also a bit of a general approach to, if possible, to reduce the use of chemical insecticides, because they tend to be quite unselective and problematic in large-scale use. It's from the beginning, it was clear that we aim at advancing or evolving current IPM strategies. We don't rely on single solutions. So we are not seeking for a silver bullet approach, which would hopefully solve all the problems in one go. This is not realistic. And as you can see here, we have also designed the project so that it consequently uses the multi-actor approach by including participatory development of strategies and also the participatory testing. So our target pests include fungi, all my seeds, but also nematodes and insects. We are focusing on France, Germany, Switzerland, Netherlands, and Poland in order to have representative areas in terms of climatic conditions, but also of the main production areas, especially Germany, Netherlands, Poland, and France, of course. We have a pragmatic approach. So we aim to substitute some candidates for substitution, and if this is not substitution isn't completely possible, we aim on replacing some of the highly problematic PPPs. Our solutions, which we bring in, which are brought in by our industry partners in SAGROPIA, all of them are based on microbial extracts and or plant metabolites, so they belong largely to the group of biocontrol agents. We have a workflow. So these alternatives, I have to say, they are in different stages of development. So some are really very close to the market and can be fully formulated and so forth, while others are a bit earlier in the development stages. And so there we aim at support the development, the product development within SAGROPIA. We also evaluate biopesticide solutions in greenhouse assays to see further potential uses if it's not yet fully exploited, so to speak, the potential. And then we have really a corner or a core part of SAGROPIA is the development and evaluation and demonstration of novel IPM management strategy. Key pests on potato and sugar beet. Of course, there is early and late blight. Late blight particularly difficult in potato, but also Colorado beetle. And then in sugar beet there is aphids, Cercospora, the fungus, which really cause major problems. Plus, is increasing emergence of known diseases, but also and pests, but also invasive ones. So this is the landscape. Constantly changing. What we have seen in these various pedoclamide 2-3 against aphids, which are quite difficult to control, 1-4 against Cercospora. And this is a landscape which is changing very rapidly due to withdrawal of active substances, but also emergence of virulent populations. Last year, we went into analyzing the current strategies and the key climates. You can see here from the top to the bottom a lines which illustrates the IPM strategies which are currently used, and I zoom in a bit here. In the case in France, where in mid-season you would have these fungicide sprays against late blight, and then later on some insecticide sprays. And you can see the bluish ones are the ones which are not yet or not candidates for substitution, but the reddish ones, they are indeed candidates for substitution. So we found in all cases situations that these candidates for substitution are still widely used in standard management practices. If you want an overview on this, then depending on the region, we have selected these various regions, as I said, depending on the pedoclimatic conditions. So maritime, humid and cold in France, for instance, but also continental dry and cold areas in Switzerland and other places. And if we looked at it, the standard STD, the standard practices, we found that in all areas, we also identified best practices, and also in these cases, the use of candidates for substitution is applied. Only in very rare occasions we see situations where the current practice, even the best practice, is not dependent at the moment on the use of these candidates for substitution. It's actually, as we go in the project, we see that it's really quite alarming how quickly the landscape changes and how quickly resistant populations, pest and disease populations are emerging. This affects the standard practices as well. So we saw that one IPM strategy which was standard last year or the previous year in the Netherlands, it doesn't work anymore properly, so it already had to be adapted to cope with resistant populations in Phytophthora. So where do our SAGROPIA solutions fit in the IPM strategies? They are actually targeted at this level in the pyramid, in the IPM pyramid. So it's a non-chemical control which should complement or can be complemented with chemical control options if needed, and this is in our case certainly the case. We have 2 work packages addressing these IPM strategies in practice, so on farms. So there is one is dedicated to the development of the IPM strategies. It's running, the first year is this year, and it will last for 3 years in these various countries. And there we have set up the experiment such that we do compare the mainstream standard practice as a reference with 3 different SAGROPIA IPM strategies which we are evaluating or proposing, which should reduce the replace or reduce the candidates for substitute in these management strategies to an expected reduction of the PPPs. And then we also have the current best practices in this given region as an additional benchmark. And it's designed such that the SAGROPIA IPM strategies, they have different levels of ambition, from having a slight reduction of candidates for substitution by, say, up to 50%, up to a full replacement. And which ones ambition. Same applies, of course, as well to sugar beet. Those strategies we AIT from Anna Babic, who does the administrative coordination of the project, and I'm leading work package 3. But of course, all of this is teamwork. And with this, I thank you for your attention.
Thank you very much, Dr. Tam, for your presentation. I don't see any questions for now, but as we said previously, if any questions come up, we will answer them at the end. So now I will ask Dr. Pasquale Saldarelli to take the floor for the BIOVEXO presentation.
Good morning. Do you see my screen?
Yes, we can see it. Thank you.
Okay, so I can start. Just
Yes.
a moment. Let me see. Okay. BIOVEXO is good morning, everyone. BIOVEXO is a real-world project dealing with big problems that the epidemic of Xylella fastidiosa in Apulia, in my region, but also with other outcome of Xylella fastidiosa in Spain. For this bacterium that is transmitted by vector, Philaenus spumarius, that you can see here, there are no available solution, and currently the only way to control this bacterium is to manage the vector population by chemical or mechanical activities and removal of inocula in the forefront of infection. It's a very problematic bacterium because it lives it replicates in the xylem, in the plant xylem. So the first problem is how to reach the bacterium with every solution, either chemical or biological. BIOVEXO is as I said, is a real world project because it deals with a set of activities that you can see here in the 5 work package that are mainly carried out in the field and also in the companies that produce the biopesticides. We moved from now it's almost at the end of the project, at the end of the year. We moved from a set of X biopesticides and V biopesticides, where X and V means against Xylella and against the vector, so bioinsecticides in this case, that were tested in a small scale, but in the field, in the work package 1. The work package 2 dealt with the optimization of the production of these biopesticides that are, I remember that is a microbial, have a microbial origin, are plant extracts from waste, and so they are all biological, they all have all a biological origin. There is a part, a work package that deals also with the toxicity, the evaluation of toxicity of the selected biopesticides, a work package 3 that deal with the production, mass production of these biopesticides that the company that belong, that participates to the project, you can see here Domka, Globachem, they are able to produce in a big amount and also to formulate for a practical field use. And now we are in the work package 4, the large-scale field trials, on which I will focus. Work package 5 also studied the mode of action of these biopesticides, while work package 6 deal with product sustainability and, as I said, toxicity. And then there are dissemination and management. I will go directly to the focus, to the work package 4, the field trials. This is a field trials. We have now 4 field trials ongoing. This is one on 120 olives of a susceptible cultivars to Xylella fastidiosa. You can see here the symptoms, and you can and they after a selection of the biopesticides, we are now working with 2 biopesticides against Xylella. One which is a microbial compounds which is applied by a foliar spray, and the other which is applied by drip irrigation, which is a plant extracts. These are these trials on this big tree of 30 years old started in February 22, and they are regularly assessed every 6 months. When I say regularly assessed, it means that every 6 months we go in the field, observe symptoms on a scale from 0 to 5, and also sample 3 by 3 to evaluate the population of Xylella fastidiosa by quantitative PCR analysis, the classical diagnostic test that is done to detect Xylella fastidiosa in plant tissue. So these are the results of 8 assessments throughout the time, and you can see that we have now 2 biopesticides that I indicate, biopesticides 1 and 2, that show not a clear reduction of Xylella of the Xylella population on these treated plants. While from the point of mitigation of symptoms, we observe a better performance of X biopesticides, 1, even if it is not exactly significant. This is area under disease progress curve, which means a cumulative evaluation of the symptoms throughout the time from February 22 to October 24. This is another field that we have ongoing. This is very interesting to me because it's a field that was planted by us in the frame of BioVexo. You can see now, this is a photo of the last the end of the 24, and you can see the size of the plants. They were planted in 2020. And again, what we are going to observe here is the results of the 6 assessment from March 23 to October 2024. Field trials with Xylella for Xylella fastidiosa, I should point that is a long process, because if you rely on natural infection, natural infection, because we are in the epidemic area, you need at least 2 years to get the first plant infected. Visible. You can see here we planted in 2020, and we observed the first infected plants by quantitative PCR in October 22. After the evaluation of the population of Xylella fastidiosa by quantitative PCR, the upper graph in these slides show that there is a lower population of Xylella in the X biopesticides treated plants. But what is more interesting for us is that we observe a reduction of symptoms on these X biopesticides 1 treated plants. Also with the plants treated with the number 2, but the best performance was observed with the microbial compounds. This reduction was significant by statistical analysis, and we observe, and we can evaluate a reduction of symptoms of 37% or 14% respectively with biopesticides 1 and biopesticides 2. We have not this activity in BioVexo, but I introduce this additional activity to have, I would say, a parallel evaluation assessment of the benefit, possible benefit, efficacy of the biopesticides that we tested. What we observed in our experience with Xylella was that the plants olives infected by Xylella suffer a drought stress. If a plant suffers a drought stress, it tends to close the stomata to avoid the loss of water. What we observed, taking these measurements of stomata conductance by eporometers in the field, in that field that I showed before, we observed that the X biopesticides 1, the plants treated with the X biopesticides 1 suffer a less stress drought stress caused by Xylella fastidiosa. So they better manage the stress induced by the bacterium. In parallel, if a plant closes the stomata, it's known that it tends to increase the temperature, because there is no loss of water, so there is no loss of temperature. So in parallel, we also are evaluating the stress, the rate of stress of plants treated with the X biopesticides, as you can see here in this example. And what we observed is that indeed there is we did several evaluations starting from 4-5 plants per treatment. I remember I told you that we have 120 plants in the fields. So each plant is we took photo from from each plants. Initially from 5 plants, and then we did other assessment on 10 October and 24, and then we observed that really the temperature of the plants treated with X biopesticides 1 was lower. The plants was colder than the untreated plants. So that means that the plants stay in a best condition, does not suffer the drought stress imposed by the bacterium. From this temperature measure of temperature, we can extract a crop water stress index that tells us that plants treated with biopesticides are less stressed than untreated plants. In parallel, we have, in addition, other 2 field trials with plants that we planted in the frame of BioVexo. To evaluate a possible IPM strategy for managing Xylella infections. In these trials, which are which contain 432 olives each. We have 2 trials, one in Squinzano and the other in Francavilla, of course, organized in randomized plots. We treat we treat also the vector. We treat the vector when the vector is in its juveniles stage with V biopesticides. We previously evaluated the efficacy of these V biopesticides in small biopesticides against the vector. We evaluated the efficacy of these biopesticides in small-scale trials, and we found that it's efficient in controlling the juveniles and the adults of Philenus spumarius. The evaluation is made by counting the nymphs and the spittle on the weeds. And in parallel, we also treat the adults with the biopesticides, the V biopesticides, and in parallel we have a chemical for comparison, a chemical biopesticides, V biopesticides against the vector, which is acetamiprid, which is allowed for treating Philenus spumarius in Apulia. All the plants, the application against the adults are made according to the sides, to the flight of Philenus spumarius. As you can see from the photos, we have sticky traps, and we apply the V biopesticides according to the population size of the Philenus spumarius, of the vector in the fields. We also have curative trials against Xylella fastidiosa in Mallorca. These are carried out on almonds, which is, you can see here some symptoms of Xylella affected almonds. Unfortunately, this trial started late in 2023, and we don't have more consistent data about the outcome of this, the use of these biopesticides on almonds. We will have this data for the end of the project. So the achievements of BIOVEXO up to now is that we selected 2 biopesticides against Xylella and 1 against the vector. We are also studying the mechanism of action. I did not talk about some transcriptome studies that we are carrying out and we are going to publish in the next future, but also we are studying the microbiome of infected plants. We also have data about the toxicity of the biopesticides we selected, and there also the sustainability by studying the life cycle of the production of these biopesticides. We defined a mode and frequency of application of these products. We observed the slowdowns of the symptoms, as you can see on the canopy of olives on plants treated with the X biopesticides. We also have observed the reduction of the juvenile and adult population of Philenus spumarius using the V biopesticides, and we designed a strategy, an integrated strategy of management of the disease based on these biopesticides. The impact of BIOVEXO is, our hope is to restore the yields of olive oil, olive production in my region is take account the 45% of olive production in Italy, so to restore the yields, to provide some options for organic farming, because now you only rely on chemical to control acetamiprid to control the population of the vector. Currently, indeed, the situation is that we had in the buffer area the possibility to eradicate and use synthetic insecticides, while in the epidemic area, we in other projects we selected some resistant cultivars that are allowed to be planted. And that's all from my side. Thank you.
Thank you very much, Dr. Soldarelli, for this insightful presentation. I don't see any questions for now in the chat, but let's give some time. Some questions have already been added to the chat, but they have been already answered by Professor Vontas. So let's wait a bit to see if any question come up, and then we will proceed to the next presentation, the presentation of Vertigation project by Dr. Kumar Vasudevan.
Yeah.
Okay, I don't see any questions for now, so please, the floor is yours. We can see your screen.
Okay. Do you see in the presentation mode?
Yes.
Okay, perfect.
Thank you.
Hello, everyone. My name is Kumar Vasudevan. I work at KU Leuven in Belgium in the Vertigation project. So today I'm here to represent the Vertigation project. This Vertigation project involves partners mostly from European countries, and the consortium include partners from universities, research institutions, and also private companies with a good network of people focusing on multiple aspects and work packages of this project. And I don't know why I cannot go. Okay. Yeah, the main project objectives are to create co-creation with the stakeholders to develop innovative diagnostic tools for detection of plant viruses and to study the plant virus vector interactions and to see the viral disease spread or prediction under climate change conditions by studying how the virus mutate under elevated temperature, for example, and then integrated vector virus management, and also about train the value chain. Yeah, and the project focused on two major plant viruses. One is tomato brown rugose fruit virus, which is an RNA virus, and it is mechanically transmitted virus that spread through gardening tools and through soil, water, and seed. And this virus also stays active, like it can stay infectious for a longer period of time. So this requires a desanitization procedure before the next crop cycle. Mainly, this affects the indoor greenhouse crops, such as tomatoes, when it's produced under a closed condition. And the second virus is tomato leaf curl New Delhi virus, which is a DNA virus, and it is transmitted by whitefly vector. And the project focused on two major crops, tomatoes and cucurbits. Today, I will talk mostly about the second virus, which is tomato leaf curl New Delhi virus, because it's transmitted by whitefly, and we are working on biopesticides to control the whitefly. So tomato leaf curl New Delhi virus is prevalent in Apo region in the northern part of Africa and also in Asia. And it affects many economically important crops such as tomatoes, potato, cucurbits, and eggplant, pepper, and so on. And like I said, it is transmitted by Bemisia tabaci, which is whitefly. And in this project, we are mainly working on biopesticides that in this work package, sorry, only on this work package, we work on the biopesticides to control the whiteflies. And we first started with nearly 10 biopesticide candidates under lab condition. And this is the overview of the testing under lab condition, where we studied the adults and the egg stage together, and the nympho stage 1 and 2 together, and the nympho stage 3 and 4 together. So to have, because the efficiency of biopesticides in general can be lower than synthetic pesticides. So we want to assess the efficiency on various developmental stages of the whitefly. And we did several treatments, untreated and for a control, a negative control, which is formulation with just additives, and a positive control, which will be one of the synthetic pesticide commonly used, and also a positive control representing a biopesticide, such as a cytaractin, and the test treatment, which is our biopesticide candidate. And like I mentioned, the biopesticide reference product we used was a cytaractin, oikos. And for synthetic candidates, we used closure, which is sulfoxaflor, or mospilon, which is acetamiprid. So we mostly used either this or that, not both, meaning one of the biopesticide and one of the synthetic pesticide. And after we did our lab testing in K. Leuven, we chose top 4 candidates, and we sent those best performing candidates to our project partners for further testing. So here is an example from results from Germany, and this was done under greenhouse condition. And here 4 formulations were tested with 4 replicates, and the Mediterranean type of whitefly was used in this, and 2 spray applications were performed. And the trial was conducted following EPO standards. And all of the 4 test formulations showed a comparable efficiency to the reference products. And in some of cases, these test formulations showed, in some case, higher efficiency than the reference product. And similarly, we also tested candidates in Spain by our project partner from Teknova. And in this trial, 3 formulations were tested, and with 4 replicates again. Each plot is 1 replicate, so 4 replicates of each treatment. And in this case, locally sourced wildlife population was used, and 2 spray applications were performed. And again, EPO standard was followed in this trial as well. In addition to testing on whitefly, this trial also included assessment on 2 beneficial insects and 1 pollinator. And the efficiency of this trial was again comparable to the reference products. And more importantly, none of the test products showed any toxicity on any of the beneficial insects that were tested in this trial. And the last trial we conducted so far was in Italy, the field trial on tomatoes. And in this case, we tested only 3 test formulations due to space, and 3 replicates were performed. Here again, 1 treatment is 1 plot, and locally sourced whitefly population was used in this trial. And we did 4 applications in this trial, and again, we followed EPO standard, and 2 of the beneficial insects were used. We didn't use pollinators because there was no flowering yet at that time. So all the test formulations again showed a comparable efficiency to our reference products. And once again, like we observed in Spain, we didn't see any toxicity on both the beneficial insects that were tested in this trial. So the next step, we plan to do IPM trial, which includes synthetic pesticides, and meaning 1 spray application with synthetic pesticide followed by with the biopesticides from our trials. So this is to see if we can reduce the amount of synthetic pesticide and also to give a better outcome, because we believe all the biopesticides that we are testing are contact pesticides, so the efficiency overall can be lower than the synthetic pesticide, so because it cannot stay longer, the efficiency. So this is why we want to see if it can be a combination of synthetic and biopesticide, and also to perform a multi-location trial, and further to improve the formulations in more in the product stage. And the last slide, it's about TOBRV, the first virus which I mentioned, which is an RNA virus and mechanically transmitted. So this, I'm not working on this project, but one of our project partners are working on this at ECM. And here it's about cross-protection strategy, where a milder variant of virus will be tested as a first vaccination for the plants, so the plant immunity gets triggered and will be prepared for infection from more severe virus of the same category. So this is the approach here. They are testing to see if we can use a mutated, less virulent strain of TOBRV, and if we can use this approach as a vaccination mechanism to induce natural resistance in the plant. So this is one of the IPM approach in this project. And that's it from me. Thank you.
Thank you very much, Dr. Vasudevan, for your presentation. It was indeed very detailed. For the time being, I don't see any new question. One question was added, but it was just answered by Dr. Saldarelli. So let's give some more time if any questions come up. And just to inform you now about the following steps in our agenda, we just completed the project spotlights sessions, the presentation of the 4 projects in the context of which today's webinar is organized. And after that, we will proceed with a short break of 10 minutes, and then we will have the expert roundtable. So again, I don't see any new questions. Maybe, yes. So if any questions come up, we will answer them at the end. So now I think we can have our 10 minutes break. We can close our cameras and microphones, and we will be back in 10 minutes from now to continue with the expert roundtable. Thank you all.
Yes, welcome back, everybody. Can you hear me okay? I guess. Angeliki, can you hear? Can you hear?
Yes.
Yes. Perfect.
Might be a bit delayed.
Mhm. Okay, so now we'll be starting our roundtable discussion. I would like to thank the presenters because these were really insightful presentations, and we learned quite a bit about biopesticides and what all these different projects are actually aiming at, which is overall making the environment more sustainable, agriculture more sustainable, and providing healthier food also for all of us. So I would like to dive into the questions right away, as we have a total of 40 minutes, and I have prepared like about 5 questions. So it's around 8 minutes per question. So let's see how well we do with this. At the end of this panel discussion, there will be time for the audience to ask questions. So for now, we just start with our presenters and speakers. The very first question is a bit general question about how do you envision, and you can see the questions here on the screen, how do you envision the future of integrated pest management for the protection of ecosystem and human health evolving over the next decade? So I would like to start with maybe the same list of as the speakers. Let's start with Professor Vontas. Then we'll move on to Dr. Tam, and then Dr. Saldarelli, and last Dr. Vasudevan. Thank you. So Dr. Vontas, please tell us.
Thank you. So in my opinion, the future of integrated pest management needs to be on an evidence-based, scientific evidence. There are not that many solutions in my opinion. There are no magic bullets that, for example, a new green chemical or a new RNA pesticide or something will provide solutions for everything. I don't think this is likely to happen. We need to rely more on sophisticated approaches, scientific evidence, integrated approaches, case-specific education, training for the farmers so that they know what they do. We go to precise plant protection approaches in line to precise medicine approaches in pharma. This is the way forward, in my opinion. For the second question.
Sorry, excuse me.
Sorry.
We will just, sorry, I know we're showing 2 questions at the same time.
Okay, the first question. Okay, I'm done.
Yeah, thank you. Thank you very much.
I think.
Dr. Tam?
Yeah, it's a good one. I think there are a few big trends there. So the availability of the chemicals, of the active substances as we know it now will decrease. That's no secret. And as a consequence of this, there will obviously be less solutions available, but there will be also the pressure of resistant strains and pests and diseases increase. And so how can it be compensated with? One strategy is, of course, to accelerate the introduction of innovations and innovative products at a very high rate, because we will need more of them, because they are by nature more selective, or tend to be more selective than the ones that are there now. We need many more, or we need many to replace each of them. So it has to accelerate, and of course it has to be registered in time and so forth. And probably at the same time there will a pressure to also to rely more on the basics of IPM, which is the use of agronomic practices, the reduction of sanitary methods, the use of resistant cultivars, and so forth. So once the availability of cheap standard solutions decreases, all these supportive and preventive activities will become relatively more attractive to be used. And these more sustainable practices will also become, in comparison, economically more viable if there is no alternative available. So if the standards are not working anymore very well. And maybe as a last point, the emergence of new diseases and pests and of invasive pests and diseases is really quite worrying. So this will also shape IPM strategies in the future quite a lot. So we will have to invent new strategies and new stuff at a really high frequency and a high pace to be in time and in place. Thank you.
Thank you, Dr. Tam. Dr. Saldarelli?
I see, I would say, a pink future, because many of the pests that we have talking about this morning are, for example, vector-borne. Many of the pests are easily transmitted, even the viruses from Kumar. So in this case, you should consider the whole strategy, the whole epidemiology of the disease. And for example, for Xylella, an integrated pest management strategy is mandatory. If you don't control the vector, you lose your gain. That's the same for many other viruses transmitted by virus. So of course, integrated pest means that we should be more precise, and of course we need to have a scientific background which base our strategy as a base for our strategy. And we need, of course, to have all the data to implement such a strategy. If we see also this strategy in the frame of a biological control, we need to develop, as we are trying to do, biopesticides to be applied in a more precise mode of application and use. So I see a mandatory future for this.
Thank you. Dr. Vasudevan, please.
Yeah. Yes, I also do believe that there is a big future for the IPM approach and more of eco-friendly solutions, and with more and more of awareness of safety issues with synthetic pesticides that you can have on ecosystem and for human and animal health. I think there is more need for safer solutions and involving not just a biopesticide, but also integrated pest management. And it involves considering the plant resistance and also application of safer chemicals, including biomolecules like RNAi or peptides and stuff. So definitely this gonna be more useful and safer options, and will be less chance of resistant development by the pests because it's not a single approach. So it's not easy for a pest to develop resistance against so many strategies when we put them together. So, and also biopesticides, if it comes from plant, for example, most of them would be expected to be contact pesticide, and it's not easy to develop resistance against a contact pesticide, but rather for a systemic pesticide where the plant, the pest is exposed to chemical in various concentration. It takes time to develop resistance, but contact pesticide reacts immediate and by suffocating the pest, for example. And there is not enough time for a pest to develop resistance against something like this. But the downside is it's going to be less effective because it's only on contact going to react, but it's not going to give long-term protection. And that's where the integrated approach is going to be promising, giving a balance of everything. So I think definitely there is a need and importance for this.
Thank you very much. Thank you. So it seems like we're all big supporters of IPM strategies and biopesticides. As expected, we have highlighted the importance of research, of evidence-based data, and also of data, and so and as well as the economic viability of many of the solutions that we are proposing. This is all very good work and very good science behind also all these new developments in agriculture and agricultural sustainability. However, there are some external influences that may affect the work that you all are doing, for example, those caused by climate change. And with this, I would like to move on to the next question and ask you, in what ways are these projects ensuring the long-term sustainability of the plant protection solutions in the face of increasing climate variability and extreme weather events? So I would like to start again, perhaps in the same order, with Dr. Vontas, please.
Yes, thank you. It is true that climate change has influence on pests, mainly arthropod pests that can migrate. For example, Spodoptera frugiperda is now a main and a very important pest in Europe, and there is another European project that kicks off in June, which specifically looks at Spodoptera frugiperda. However, I would say that sustainability seems to be relevant to those sophisticated evidence-based IPM projects. For example, approaches. For example, one important component of sophisticated IPM is the IRM, the insecticide resistant management, which means it's you make a rational use of pesticides only if needed and when needed. You have many tools, and so you can use alternative approaches in rotations or appropriate mixtures and substantially reduce the use of pesticides, which means most likely you are proceeding with a more sustainable SEM indeed. So, however, to be honest, I'm not confident at this point that we have enough alternative solutions in terms of efficiency, because of course agroecological approaches are fantastic, are sustainable, but the efficiency of these approaches when you have problems that are also fueled by the climate change, so high populations of pests or diseases, may not be enough, the efficiency, and in that case we will need more tools, which are becoming more and more difficult, particularly in Europe. Thank you.
Thank you. Dr. Tam, please.
I agree to what has been said so far. One strategy, of course, is to diversify. Diversify in the sense of using the, as I stated previously, more of the preventive strategies like varietal resistance and so forth to stabilize the system. And the other aspect of diversification would be to introduce and use carefully all new components of the pest control or disease control toolbox, and to use them wisely to prevent resistance, buildup of resistant populations, etc. Now, if we are at the individual product level, it's sometimes a bit difficult to say ex ante how high is the risk of building up of resistant populations. And I believe there it's it was said before, science-based research is absolutely crucial. So if we know the mode of action of the different techniques, tools and techniques, products we are using, then this gives us quite a good indication on how to use it wisely to preserve its efficacy over a longer period of time and to maintain the efficacy. Thank you.
Thank you very much. Dr. Saldarelli?
Yes, it's a very critical question, this one. So what we, if we refer to this project, I would say that we are learning how to use these plant protection solutions in this increasing climate variability and the extreme weather events. So I think we are accumulating a sort of background of information that will allow us to better implement an integrated pest management strategy. For example, the last summer season was very drought was very dry in our region, and dry with Xylella is a cumulative stress that makes. So we are learning how to face the combination of the two stress also in other projects. So I would say that this project allow us to accumulate experiences and background information that would allow to ensure a long-term sustainability of this plant protection solution. That's the main outcome, I think.
Yes, thank you. Dr. Vasudevan?
Yeah. So for the climate change, yes, for example, in mitigation project, we are, not me, but our project partners are studying the impact of elevated temperature on the viral mutations. So with the sequencing approach, we can see what happens to the virus when we expose to altered weather condition, whether the virus mutates into more severe one or becomes more milder variant or so. And also the impact of climate change on the insects, whether they transmit the virus more faster, more stronger, or conditions. It can still give us an idea of what could happen and how to be well prepared in advance. And another aspect of this is what kind of mutations are going to potentially cause an outbreak in a climate altered condition. So this will also in a way help with such diagnostic tools. If we get samples from as a monitoring approach, seeing the viral mutations, we can in a way predict with the help of lab studies like a potential outbreak could happen or not. And yeah, I think in future there will also be involvement of drone technology where we observe, integrate weather conditions, predictions, and targeted application of spray products that are not wasted but more efficiently applied. So so considering the weather changes that can happen locally and targeting the application of products according to that, and yeah, considering the viral changes with the elevated temperature or drought condition, in some cases, I think the virus, and when the plant is under stress condition, can probably become less virulent because it doesn't want the host to die. Maybe so that can change from virus to virus. So all these definitely involve studies first to understand what can happen under different abiotic stress conditions, and will help us to better be prepared for how to manage such situation when it happens.
Thank you. Thank you very much. So I think all these projects are helping us. The science that's been collected throughout all these 4 or 5 years within the project are helping us to understand the impacts of climate change, and we are collecting data to develop better practices, more sustainable practices. But this is definitely one important question that we need to answer over time, how climate change is impacting sustainable agriculture. So far we have discussed how scientists are working very hard to find alternative solutions to chemicals. But as we know, farmers will only implement what makes them sell it. And so it's important to understand the supply side, but also the demand side. And with this, I would like to ask the next question, which is what role do you see for public awareness and education in promoting integrated pest management practices? So as usual, in the meantime, I would like to also invite the audience to type your questions in the chat so that we would have a list of questions already prepared during the Q&A session at the end of this panel discussion. But now I would like to invite again Dr. Montes, Professor Montes, to answer this third question. Thank you.
Yep.
Thank you.
Yeah, it's extremely important role. I'm not sure, to be honest, if the structure for implementing this education and training and those demonstration fields in particular are present across Europe. For example, I know that in France, INRA is extremely well organized and provides that service to the end users. So the outcomes of the research outcomes are much closer to the end user and the innovation. In places like Greece, unfortunately, we don't have this. I mean, the respective INRA, which is Elgo Dimitra, is not so well organized. I mean, it's not supported by the government. That's the problem. So, in many occasions, universities or research organizations like Forth that I mean, take over this task. But the reality is that we are not that relevant. We are not that close to the farmers. There is a gap, there is a distance. So I don't know if additional funding should be directed towards this gap, although it is true that in this particular frame, we have made a big efforts from what I managed to capture in all projects to include such components. So in some cases, with help from experts in other places in Europe, we will also try to develop our links with the end users, for example, in Greece or in countries where this link is not obvious. So it is an extremely, this component is extremely important so that we maximize the outcomes and the utility of the outcomes that we get from the research projects. Thank you.
Thank you. Dr. Tam?
That's a tricky one because the public doesn't know much about plant protection anyway. It's very far from their daily life. So what I see as pillars to promote IPM, it's quite evident or obvious. Okay, there might be the intrinsic motivation of farmers using it or doing it because they want to improve something. And then, of course, there is the pillar of making information available to them that was raised a minute ago. So the technical information on how to use the techniques must be available to farmers. Then the transition doesn't happen on its own because quite often it takes more time or it costs more to the farmers and so forth. So policy support is also absolutely key. And if we think of the public, then you would need the support of the public if any regulations are put in place or subsidies or nudging elements from policy support. So consumers would need to understand it, why this should be put in place. so that also the farmers are not left alone, just with a higher production cost. But then the most important leverage for consumers would be over demand. As soon as retailers say we want this serious IPM produced, or if you like, organically produced foods, not something in between or something weak. If they would do this, then this would have a massive impact, of course, on the production side, because this would also strengthen the motivation of everyone involved and also shape the market, how farming is done. But the key challenge is really to bring the dry information of what a fungal pathogen is or a pest and what it means to consumers, and that's really quite a challenge.
Mm-hmm. Yes, thank you. Dr. Saldarelli?
A major role. A major role, and we experienced this because we are in the middle of an emergency in this region. So I imagine an in the middle of an epidemic, you should imagine an integrated pest management strategy on a wide area. So you cannot rely on spotted interventions, spot actions to solve the situation. So for us, it's a basic. And this has been raised, public awareness has been raised in Apulia because of the severe problems that we had with Xylella. So we regularly, also the phytosanitary services in my region have done, there is a website, for example, of the Apulian region that educate, I would say, but also inform people about the progress of the epidemics with maps, and also describe the way of managing the Xylella infection at the border in the buffer area. So public awareness and education is a critical point for important pests, for managing important pests.
Thank you. And Dr. Vasudevan?
Yeah, I'm not sure how much public is aware of IPM and new pest management strategies and integrated approaches. Not sure of how much of awareness is in there and how much more is needed. Maybe definitely it's always more awareness is always good. But I see it more like when it comes to human health, the misuse of antibiotics or the risk of antibiotic resistance are not much public or aware of, but it is regulated by, by restricting the access to antibiotics by medicinal practitioners. And so I think it's like this public, maybe they don't know about much of the risk of different kind of pesticides and the advantage of different approaches to safer approaches. But when it comes to consumer level, they probably only see as what is labeled as organic and bio. So maybe it comes to that level in addition to awareness, but also to different kind of the end product labeling. Probably that's how it's getting communicated to public. Or yeah, I'm not sure how to raise this awareness of IPM to the public. Yeah.
Yes, thank you. Certainly a challenging aspect of research and of scientists having to reach out to the public to inform them about what they're doing and how important it is. But we mentioned also that policy support is extremely relevant here. And with this, I would like to ask maybe this question, the next one, So basically this question is about the collaboration with the stakeholders, including, let's say, policymakers, for the success of these projects in particular, but also for the success of the raising awareness and informing the public about IPM strategies and sustainable agriculture. Now, because we're kind of running out of time, I would like maybe only 2 of you to answer this question. Whoever is willing to do so, you can just start speaking or raise your hand instead of going through the entire panel again. Because then we have one last question, which is more important, I believe, or perhaps not more important, but for the projects is more important. So I would like to devote a bit more time to the next question. So I don't know, whoever feels like wanting to answer this question number 4. So the importance of involving policymakers and in the success of these projects. Yes, Dr. Tam, please.
Okay, I'm trying to answer this unimportant question. Of course, the collaboration at least here is super important, and I think we have made great progress in all as is shown in all these projects, because industry is involved and we are reaching out. Something which is, however, missing, I think, is we are not sufficiently efficient in reaching out to stakeholders further down along the value chain, meaning food processors and retailers and so forth. And this, as I said a minute ago, is quite crucial if it comes to the implementation of the novel techniques. So if we can improve on this in the future, I think this would give us added value and help us to bring the techniques more rapidly into practice.
Thank you, Dr. Tam. Dr. Saldarelli, and then I see another hand up, but since we're now only working with having a discussion with the panel and the speakers, we'll keep this question for later, for the Q&A session. Thank you. So Dr. Saldarelli, please.
Crucial. It's crucial. We have experienced this throughout our experience with all the projects that we have with Xylella. Because but it was natural in our situation. It was natural to stakeholders look for research, ask for research. So we are in the opposite situation, in which we should be careful in provide solution until we don't have a real solution. That's our situation, because the status of the epidemic is so severe, so disease, that we have a lot of interaction with stakeholders. For example, in the region, in the core of epidemics, we have greenhouse established in an olive oil mill, a strong collaboration field orchards that stakeholders put at our disposal to make tests and to test new varieties and so on. So it's and of course policymakers in this scenario, they are pushed from the base, from the farmers. They are pushed to do something. That's the situation.
Thank you. So it is very important, the involvement of policymakers, and we understand that once there is an emergency, for example, the implementation of these novel techniques is extremely under stress, under pressure. And so this cooperation with the policymakers also to facilitate perhaps the approval of specific biopesticides is also extremely important. So we're now moving on to our last question, which is about common challenges that can be identified across all these projects. So NextGenBioPest, SAGROPIA, BIOVEXO, and VIRTIGATION. And how can perhaps collaborative efforts help address the issues that we're dealing with? So this last question is about, have we perhaps identified today with this round of presentations, some collaborative efforts that can help us understand the issues better. And here again, I would like to start with Dr. Vontas.
Yes, thank you. Yes, it's pretty obvious that there are common challenges at several levels, from regulatory. For example, in many of the projects that were presented, biopesticides have been proposed. For example, microbial biopesticides or mixtures of plant extracts or other things of biological origin. And as I mentioned earlier, the frame for particularly in Europe is not very clear, or for double-strand RNA pesticides. Some of those products are already in the US market, but it doesn't seem so straightforward in Europe. So, and I believe across those projects there are similar biopesticides, so we could maybe all together look at that. Also, in collaboration with Resion, which is a project targeted on this particular issue. But further than the technical issues, as mentioned in the previous questions, education, translations into policy of this kind of sophisticated, novel, not so novel, but advanced IPM approaches, which really need to be based on scientific evidence. And it's a new concept. It's not just IPM, okay, that I use several tools. It's how you use those tools, when you use these tools, and precise applications of its available control method and product. So this translation into policy and the training component, of course, require a collaboration. And again, in most of those projects, we could develop common efforts towards those directions.
Thank you, Dr. Vontas. Dr. Tam?
Actually, it refers to your previous question to some extent. But first, I think what have we in common? So the registration requirements for novel products are still quite difficult, as we all know. So we should keep up the pressure or the motivation of those involved to facilitate appropriate registration procedures, which is well on the way, but there is still plenty of room to improve in Europe. The second bit is the communication, or the communication and dialogue with the wider public. We have just discussed a minute ago that it's difficult to reach out to the common citizens because they don't know much about plant protection anyway. And unless we have a subject like Xylella, which is super important in a specific region, then you can reach out and reach to the public locally at least. But in a more generic way, I think it's difficult for individual projects to do so. So it might be worthwhile to join forces in a way to bring the whole range of activities, all these projects, in a format or in a context which is then digestible to a wider public. I think this would benefit all of us.
Yes, very good point. Dr. Saldarelli, please.
I agree with Dr. Tam. With the registration of the possible project is a difficult task. I learned this as a scientist, this new activity in the frame of BIOVEXO. I learned this. I'm learning from the companies that belong to BIOVEXOs. So that could be, as a collaborative efforts, I see many points of contacts with with vertigation, for example, because of the vector-borne disease pest that are also of interest. This so sharing information on how to manage on the epidemiology of this kind of disease could be important, could be a collaborative for us. The other interest from me, but as a molecular origin, is the use of RNA interference, the double-stranded RNA in this kind of formulates. I know that some colleagues of my institute in Torino, they are trying to also to use this against the vector of Xylella fastidiosa perhaps, and this is an interesting task that could be interesting for me.
Thank you very much. Dr. Vasudevan?
Yes, I agree with almost everyone mentioned the same thing, which is a registration of a new biocontrol product. Yeah, I also feel that's the common problem that we all face is I think there is a need for, and it is relevant to the previous question with the policymakers, and that there should be probably a fast track system for bio-based products for new generation biopesticides or biopesticide that comes from plant or any kind of biological source. So I think for now it's all treated probably in the same channel, that it has to go through the same procedure and a long process to go through that. So yeah, I think there is a need for a special channel for treating the applications that is related to biological source that comes from microbes, from plants. And yeah, I think this would help projects like this to reach the market faster, and but not compromising any safety protocols. But I think that could be a balance of following the procedure that is required for the safety, at the same time not to be way too stringent that we do for a synthetic pesticide, to speed up the process that will effectively replace the problematic synthetic pesticides with the more safer options. And I think this is the common problem that we all agree on.
Yes.
Yeah.
Thank you.
Thank you.
Sorry, did you want to continue?
No, that's it. Thank you. Yeah.
Okay, thank you so much. Yeah, so clearly the registration of the products for biocontrol is a challenge. As far as I know, the European Commission is working on simplifying this process. But it's very important also to understand this and try to find some perhaps collaboration between the projects, as has been already mentioned. So I hope this webinar helped in that. And now I think it's time to kind of conclude our panel discussion. So I would like to thank all the speakers for their very insightful answers to this varied type of questions. And I would like to open the floor to the public or the audience to ask questions. I see already 2 questions in the chat. One is regarding, you can also, if you want, just ask the question yourself. I see Stefania, sorry, I have to change my glasses because it's very, very small font. Luzzetti is asking, how many applications do you foresee with the use of DS RNA for efficacious acceptable results? And then the second one, as the farmer also would need to consider the costs. For example, how often would he need to enter his fields, petrol costs or other resources costs? So I don't know who would like to answer of the speakers. You can raise your hand or just start speaking. You can also see the questions in the chat if you need to reread it.
Yeah, I'm not sure if this question is addressed to me, but I can answer if no one else going to do that for double-strand RNA. I already answered in the chat. So when you do the field trials with double-strand RNA products, you of course also consider the cost, including frequency of applications. And it seems that the effort required is very much equivalent to chemical insecticides. So the new formulations for double-strand RNA products provide metabolic stability, so the product remains there for 7 to 10 days, depending on the application. You don't need more. I mean, maybe you need slightly more applications, but the overall cost efficiency is equivalent to chemical insecticides for the specific trials that have been conducted already against mildew and mites and Tetranicus urticae in the certain crops, okay, in grapevines.
Okay. I hope this answer was satisfactory, or Stefania, in case you can please take the floor. Dr. Vasudevan, please.
Yeah, I have a question also about double-stranded RNA. Is the efficiency works well with single target, or is it necessary to do multiple targets in one construct or in one double-stranded RNA to have multiple genes that are targeted in the same application, or does it works even with single target? Yeah, that's my question.
Is this for Dr. Vantas, right?
Yeah.
This question?
Yeah.
Yes. Please,
Yes.
Dr. Vantas.
Sorry, excuse me. I didn't get the question because I was trying to read Jose's
Sorry.
message. So can you
Yes.
please repeat the question?
Sure, no problem. So my question was, does the efficiency of double-stranded RNA works with single target, or is it necessary to involve multiple targets in the same construct?
Thank you. It's now, I mean, the trend is to involve multiple targets also to delay or avoid insecticide resistance. So this is how company proceed. They don't, and then it's more flexible, the product. But yes, multiple target is the way to go.
Yeah, thank you.
Yeah, thank you very much. Now I will take the question by Mona Jahani. Please ask your question, since I don't see it in the chat. And your hand is up. Mona, did you want to ask a question? You're muted. Okay, we'll move on to the questions on the chat. Now there is a question from Phil Elmer. Hi everyone, my question to you. You can also ask it yourself, Phil, perhaps, if you prefer. Or
yeah, sure. Yeah. Hey, look, hi everybody. Yeah, I don't think I've met most of you, but Stephen Webb kindly invited me to attend because he's been in New Zealand and scoping out new opportunities to help with some of the European applications. It was fortunate enough for us to meet with Steve. We've been working with biocontrol and induced resistance for 30 years. So our team has developed 5 registered now biological control agents. There's a lot that we've learnt about doing that process, and our latest biocontrol, which is for deployment in kiwi fruit for 2 diseases. One is a fungal disease, the other is a bacterial disease, bacterial canker, and we were able to develop that in 6 years. And you know, I think there's a lot we've learned from that, and we'd be very happy to help you in your project in however way that you see most fit. So just get in touch with me, and I'll put my email in the chat if that's helpful. Thank you.
Yes, thank you, Phil. Please put your email in the chat, and that's definitely helpful. So I see a comment from Jose Cavallo, and it's insights from the industry. I'm not sure I need to read it out loud, as you can read it yourself, I guess. I would like to spare some time for more questions. I see that Mona is not able to unmute herself. Perhaps you
now I guess I will.
Perfect.
Now I can. Yeah.
Perfect.
No.
Go ahead.
Hello, thank you. I have my point is toward more policy and advocacy, because I guess that it's very important that like we as like all the people who are developing this innovation are less involved in policy and advocacy of these products. And then as long as you don't have any good regulatory market for this in Europe, you have products, but then you have to go to Brazil then to have this product in the market. So you have your innovation, but you cannot put it on the market. So I guess the role of policy and to make a regulatory more open in the Europe market is even now to me is more important than the innovation per se. So it's just my own opinion of working with farmers and yeah, seeing how hard it is to put some of these products. Most of these biopesticides are very hard to put it on the Europe market. So
who would like to answer or address this comment by Mona regarding the regulations and the registrations of new bioproducts? That's what we discussed already. Yes, it's definitely not an easy one in the European Union. But she's saying that maybe some of these products are then registered in Brazil, if I understood correctly.
yeah, I guess some of them, because it's very easy to put many
Yes.
of them. Like,
Yeah.
yeah.
Did you have any experience with that? Maybe any of the speakers?
It's far more difficult to register such products in Europe, my experience, and that's also because its regulatory is far more strict. Sometimes not fully justified, to be honest, but this is unfortunately the situation. I also, if I can say a word for José's comment about specificity required, I mean, the Europe is requesting products extremely specific to make sure there's no side effects on pollinators or any other beneficial organisms, and so ideally to have a pesticide for a specific pest. This is very difficult, and I fully agree with José that it's not attractive for companies because the market is very small. So companies are not interested. They prefer to go with mode of actions with broader efficiency and sell in and register them in South America and Southeast Asia, where the market is much bigger. So there are serious regulatory issues, unfortunately. So all this we need also, I mean, we need to develop procedures, frameworks, things like that, but also it's important that people like us, researchers, also have a voice, and it's not only the environment that matters, but also the practical solutions. Of course, we are all interested and we all work towards reducing pesticides residues in the environment, but we also need to think of practical solutions, which is because that's also important to in order to sustain crop production.
Yes, thank you very much, Dr. Vontas. I don't see any other questions in the chat, but if anyone wants to now ask maybe the last very last question, we still have maybe a couple of minutes. But if there are no more questions, then I would like to thank again the speakers and for your time and for a really insightful discussion. I think this webinar has really helped us to connect these different projects that are working in similar field. So I hope this will continue also in the future, and this collaboration perhaps. Now I would like to pass the floor back to Angeliki for the concluding remarks. There is one more question. This we already addressed. Yes, thank you very much. Angeliki, the floor is yours.
Thank you. So from my side, I would like again to thank you all for attending today's webinar. As it was mentioned earlier, collaboration between stakeholders and the Horizon Europe project is very important. So for this reason, I strongly encourage all participants to check these project websites to find the contact details. All of us, we are open. If any questions, any suggestions, any insights on the actual needs of the farmers, needs for policymakers, market needs, etc. So please visit our websites. There you will find also several insights in the field of pest management and sustainability. The recording of this webinar will be available in the next days through our social media of all projects. So you will be able to rewatch it in case you want, or even to share it with your network. And I think that now we can conclude this insightful webinar. Again, thank you all. Thank you to the participants and to the speakers. And we hope that we see you again in a future webinar.
Thank you.
Thank you.
Thank you very much. Bye-bye.
Thank you.
Thank
Have a
you.
good
Bye.
day.
Bye.
Thank you.
Bye.
Thanks, everybody.
Thanks. Thank you. Bye.